The convergence of artificial intelligence, computational biology, and messenger RNA engineering offers transformative potential in therapeutic design. This compendium presents 5 computational studies addressing Alzheimer's, autoimmune diseases, opioid harm reduction, lysosomal storage disorders, and chronic wound management. Each study employs advanced algorithms for messenger RNA sequence design, immunogenicity profiling, and systems biology modeling, showcasing artificial intelligence's role in optimizing therapeutic strategies. The compendium explores artificial intelligence and computational biology's role in messenger RNA therapeutics, addressing Alzheimer's, autoimmunity, opioid harm, Hunter syndrome, and wound management. Key methodologies include transformer-based algorithms, RNA structure prediction, and CRISPR analysis. Results show promising computational efficiency, reduced immunogenicity, and potential cost savings, highlighting the need for experimental validation to confirm these findings. Artificial intelligence-driven messenger RNA therapeutics leverage codon optimization and computational vaccinology to address complex diseases like Alzheimer's and autoimmune disorders. Techniques such as Vienna RNA for RNA structure prediction and CRISPR In-Silico validation enhance precision. Innovative approaches include fentanyl sequestration and magnesium oxide nanoparticle hydrogels for chronic wound management, showcasing the potential of artificial intelligence in drug design. Study I explores AI-driven computational modeling for neuroprotective protein systems targeting Alzheimer's disease. It details methods, multi-layer validation, and results, highlighting limitations and future directions. Study 2 focuses on an AI-engineered messenger RNA tolerance vaccine platform for autoimmune diseases, discussing methods, results, and conclusions. Study 3 presents a 2-phase approach to reduce central nervous system fentanyl exposure, involving protein binder immunotherapy and messenger RNA-encoded protein expression. The Consolidated References section compiles all cited works, providing a comprehensive resource for further exploration of computational biology and artificial intelligence-driven messenger RNA therapeutics. This scientific index categorizes key topics, facilitating efficient navigation through the research compendium. These tools are essential for researchers seeking to delve deeper into the methodologies and findings presented. Alzheimer's disease affects millions, with projections indicating a rise to 13 million cases in the United States by 2050. Current treatments offer limited symptomatic relief. mRNA therapeutics, inspired by COVID-19 vaccine success, present a promising multi-target strategy for neurodegenerative diseases, focusing on protein systems like neprilysin and insulin-degrading enzyme for potential therapeutic breakthroughs. Insulin-degrading enzyme, IDE, and neprilysin are crucial for amyloid beta degradation, with declines linked to amyloid buildup. Brain-derived neurotrophic factor and nerve growth factor are vital for neuronal survival and synaptic plasticity, both impaired in Alzheimer's. Dopamine receptor D1 modulates cognitive circuits, becoming dysregulated in Alzheimer's. These targets form a multi-pathway intervention strategy for therapeutic development. AI-driven messenger RNA optimization employs transformer-based models to enhance translational efficiency. Codon adaptation index targets greater than 0.80 using brain-specific codon tables. Guanine cytosine content is optimized to 50-55% for stability. 5VE untranslated region engineering maximizes ribosomal engagement with optimized Kozak sequences. Secondary structure is refined to minimize inhibitory formations, ensuring efficient translation initiation. Systems proteostasis modeling utilizes NetworkX for protein interaction networks, analyzing betweenness centrality and degree distribution. Amyloid beta clearance is modeled with Michaelis-Menten kinetics, using scipy.integrate.odient for numerical solutions. Enhancement factors are derived from messenger RNA expression changes, influencing neprilysin and insulin-degrading enzyme activity on amyloid beta substrates, crucial for Alzheimer's therapeutic strategies. The validation engine class in Python evaluates messenger RNA constructs using metrics like guanine cytosine content, codon adaptation index, and Kozak sequence presence. Efficiency scores are calculated with weighted metrics, ensuring constructs meet the 0.75 threshold for experimental validation. This empirical threshold aligns with in vitro translational outputs, guiding prioritization in experimental pipelines. In-Silico optimization significantly reduced molecular weights of target proteins, enhancing therapeutic potential. Brain-derived neurotrophic factor and nerve growth factor achieved over 50% reduction by removing prodomains, retaining active domains for receptor engagement. neprilysin and insulin-degrading enzyme maintained catalytic sites, reducing weight by 22%. Dopamine receptor D1 conserved ligand binding, achieving 12.2% reduction. All constructs surpassed efficiency thresholds, with brain-derived neurotrophic factor and nerve growth factor showing highest scores due to optimized sequences. Network analysis reveals NEP and IDE as key nodes in Alzheimer's AV clearance, with BDNF showing strong interactions with NGF and DRD1. Simulations predict NEP-SIDE combination achieves 78% AB reduction, enhancing cognitive benefits. These theoretical outcomes, based on kinetic modeling, highlight the potential of multi-target mRNA therapeutics, pending experimental validation. The multi-target messenger RNA approach addresses Alzheimer's as a systems disease, enhancing amyloid beta clearance through neprilysin and insulin-degrading enzyme synergy. The validation engine fills a critical gap by providing standardized metrics for messenger RNA designs, facilitating comparisons. Molecular weight reduction offers immunotherapeutic benefits, reducing antibody formation risk and manufacturing costs, while improving mucosal permeability for central nervous system targeting. Functional domain conservation analysis leverages sequence homology and structural data to estimate activity retention, though allosteric effects may be underestimated. The Computational Safety Index is an algorithmic estimate, not a substitute for experimental evaluation. Future directions include in vitro transfection efficacy, protein expression quantification, and functional activity assays to validate computational predictions. Autoimmune diseases affect 5 to 8% of the global population, driven by immune dysregulation against self-tissues. Traditional treatments use broad immunosuppression, risking infections and relapses. This study proposes an artificial intelligence-engineered messenger RNA tolerance vaccine, re-educating immune responses by presenting self-antigens in a tolerogenic context, aiming to induce T-cell anergy and expand regulatory T-cells, restoring immune balance without systemic suppression. Cardiac autoantigens MYH6, TNNI3, and TTN were selected for tolerance vaccine design. Epitope selection utilized AlphaFold 2 for surface-exposed regions and transformer models for major histocompatibility complex class II binding affinity. Artificial intelligence-guided messenger RNA design optimized constructs for expression and minimized immune activation. In-Silico clustered regularly interspaced, short palindromic repeats. Cas9 validation ensured genomic safety, advancing constructs with zero off-target cleavage events. AI-optimized DNA sequences for Escherichia coli expression were analyzed using the PET28A plus vector under T7 promoter control. Predicted yields of 1 to 5 mg per liter soluble protein were estimated. Purification involves nickel nitrilotriacetic acid chromatography and phosphate-buffered saline desalting, achieving greater than 95% purity. Computational characterization includes liquid chromatography mass spectrometry identity verification and endotoxin removal feasibility, ensuring high-quality protein production for therapeutic applications. The AI-engineered messenger RNA tolerance vaccine framework innovatively combines synthetic biology with immune pedagogy, re-educating immune networks rather than suppressing them. This approach leverages adaptive immunity's plasticity, using computational design to restore self-tolerance. Virtual clustered regularly interspaced short palindromic repeats. Safety screening enhances biosafety, potentially reducing animal testing, aligning with ethical research principles. The opioid crisis, exacerbated by fentanyl's potency and rapid central nervous system penetration, demands innovative solutions. This study introduces a peripheral sequestration strategy using engineered protein binders to capture fentanyl in the bloodstream, preventing central nervous system entry. This adjunctive approach complements existing medication-assisted treatment therapies, aiming to reduce overdose risk without replacing current treatments. The drug-protein complex formed is too large and insufficiently lipophilic to cross the blood-brain barrier, reducing fentanyl's central nervous system availability. Protein scaffolds like nanobodies, DARPins, and affibodies are evaluated for their binding affinity, stability, and scalability. Peripheral sequestration avoids opioid withdrawal and is compatible with medication-assisted treatment, offering a dose-dependent protection window. Key pharmacokinetic parameters include circulating half-life, volume of distribution, and clearance pathways. Strategies like pegylation and Fc fusion may extend persistence but require immunogenicity monitoring. Safety monitoring in Phase 1 involves assessing injection site reactions, systemic adverse events, and immunogenicity through anti-drug antibody detection. Primary endpoints focus on maximum tolerated dose, pharmacokinetic characterization, and proof of concept for binder efficacy. The mRNA construct for Phase 2 includes codon reoptimization for human translation, M1-Su substitution to reduce immune activation, and optimized 5C UTR and 3C UTR for stability. It features a signal sequence for protein secretion and Cap1 structure for translational efficiency. LNP formulation focuses on hepatotropic biodistribution, optimal particle size, and pegylation to enhance circulation half-life. The ethical framework for addiction medicine emphasizes patient autonomy, informed consent, and equitable access. Critical technical limitations include the need for high-affinity protein binders and cross-reactivity with fentanyl analogs. Regulatory pathways involve Food and Drug Administration biologics, requiring investigational new drug applications and biologics license application submissions. These considerations are crucial for advancing messenger RNA therapeutics in addiction treatment. Hunter syndrome, or MPS II, is a rare X-linked disorder caused by IDS gene mutations, leading to GAG accumulation and multi-organ pathology. Current enzyme replacement therapy, while beneficial, fails to address neurocognitive symptoms and is costly. Messenger RNA-based therapy offers a promising alternative, potentially improving IDS distribution and reducing treatment burdens. The lead messenger RNA construct, variant E, enhances iduronate 2-sulfatase protein expression through a meticulously engineered architecture. It features a Cap1 structure for translation efficiency, an optimized 5C untranslated region for high hepatocyte translation, and a stabilized 3-size untranslated region for extended messenger RNA half-life. Chemical modifications, including N1-methylpseudoridine and 5-methylcitidine, reduce immune activation, achieving a 160% increase in predicted expression. The codon optimization algorithm utilized a multi-algorithm consensus approach, enhancing the native IDS sequence with tools like JCAT and GenSmart-TM. This included targeting GC content, avoiding restriction sites, and eliminating RNA instability motifs. The AI model, trained on 12,000 high-expression sequences, achieved an R2 of 0.78, validating its utility for construct ranking. The pharmacoeconomic analysis reveals significant cost reductions with messenger RNA therapy compared to current enzyme replacement therapy. Weekly dosing reduces lifetime costs by 95.4%, while monthly dosing achieves a 98.9% reduction. These projections, however, are theoretical and require validation against real-world good manufacturing practice production data, considering potential cost variations due to manufacturing scale and regulatory factors. Experimental validation is crucial for computational messenger RNA therapy models. Key studies include in vitro messenger RNA expression in HEK293T and HeLa cells, iduronate 2-sulfatase enzyme activity restoration in Hunter syndrome fibroblasts, and iduronate 2-sulfatase knockout mouse model efficacy. Comprehensive immunogenicity and pharmacokinetic assessments are essential, alongside long-term safety evaluations to ensure therapeutic viability. Chronic wounds, affecting over 6.5 million in the United States annually, are hindered by biofilms that resist healing. These biofilms, composed of bacteria in a protective matrix, impede immune responses and antibiotic efficacy. Systemic antibiotics struggle due to poor vascular perfusion and biofilm barriers, necessitating local, non-antibiotic strategies for effective management. Magnesium oxide nanoparticles, MgO-NPs, offer a novel approach to biofilm disruption in chronic wounds. Their antimicrobial action involves alkaline stress, reactive oxygen species production, and membrane interaction, reducing resistance development. Injectable thermogelling hydrogels, utilizing thermoreversible polymers, enable precise delivery into wound cavities, transitioning from liquid to gel at body temperature, ensuring effective biofilm penetration and sustained antimicrobial activity. The magnesium oxide nanoparticle-loaded thermogelling hydrogel system is engineered for optimal wound application. Key parameters include a gelation temperature of 30 to 35 degrees Celsius, ensuring rapid gelation at tissue temperature, and a storage modulus of 1,000 pascals for structural integrity. The system targets a 5 to 7 day magnesium oxide nanoparticle release, maintaining antimicrobial efficacy while supporting wound healing through moisture balance and fibroblast infiltration. The magnesium oxide nanoparticle thermogelling hydrogel system offers a novel approach to chronic wound biofilm management. It ensures conformal delivery, sustained antimicrobial release, and biodegradability. Key translational challenges include nanoparticle surface optimization, in vivo biocompatibility testing, regulatory compliance, and good manufacturing practice manufacturing scale-up. Addressing these will be crucial for advancing this promising technology. All computational methodologies, AI-optimized mRNA sequences, and pharmacoeconomic analysis parameters are freely available under Creative Commons Attribution 4.0 licensing. Access the complete source code and sequence data via Alkalili BioAI Limited Liability Company's GitHub and Zenodo. This research is entirely computational, requiring no ethical approvals, and findings need experimental validation before any application. The research compendium by Alkalili BioAI LLC emphasizes that no commercialization activities were pursued at publication. The author holds no financial interests in the technologies discussed, ensuring unbiased research. This work, released under Creative Commons licensing, remains purely computational, requiring experimental validation for any practical application. N1-methylpseudouridine incorporated messenger RNA enhances protein expression and reduces immunogenicity compared to pseudouridine incorporated messenger RNA, as demonstrated by Andres et al., 2015. This advancement is crucial for messenger RNA therapeutics, offering improved stability and efficacy in mammalian systems. Such modifications are pivotal in optimizing messenger RNA-based treatments, ensuring better therapeutic outcomes and minimizing immune responses. The compendium highlights the role of messenger RNA vaccines in modern vaccinology, emphasizing their potential in therapeutic applications. It discusses the amyloid hypothesis in Alzheimer's, exploring amyloid beta-degrading enzymes, and the therapeutic promise of brain-derived neurotrophic factor. The document also covers the codon adaptation index for optimizing messenger RNA sequences, and the use of linear design for efficient messenger RNA sequence design. The scientific index highlights key terms like amyloid beta, a primary Alzheimer's substrate targeted by neprilysin insulin-degrading enzymes, and alpha-fold 2, used for protein structure prediction. It also covers messenger RNA optimization metrics like codon adaptation index and minimum free energy, crucial for enhancing therapeutic efficacy. Additionally, it addresses delivery systems like lipid nanoparticles for messenger RNA therapeutics. N1-methylpseudouridine, M1C, reduces innate immune activation in mRNA therapeutics. Neprilysin, NEP, is a key enzyme for degrading amyloid beta, with an mRNA efficiency of 0.823. NET-MHC-PAN 4.1 predicts MHC-I epitopes for immunogenicity. Peripheral sequestration targets fentanyl in opioid use disorder. Transformer neural networks optimize mRNA sequences, enhancing therapeutic efficacy.